Hi, my name is Anne McGinnis, and for Unit 2, I'll be discussing Case 2 and Case 3. So for Case 2, an individual has a large glioblastoma tumor with areas of necrosis within the tumor in the brain. A PET scan was taken of this individual after administration of fluorescently labeled D-glucose. The PET scan showed that the areas in which the tumor was present was more lit up, indicating a higher concentration of glucose. One can infer from this PET scan result that when compared to normal tissue, glucose uptake is higher in the tumor cells. A tumor's microenvironment is often hypoxic and nutrient poor due to it being an area with poor blood supply. In order to survive and further proliferate, tumor cells must adapt in order to have the ability to obtain the nutrients needed to do so. One of these adaptations is the Warburg effect, which explains that cancer cells take up large amounts of glucose and metabolizes it via glycolysis to lactic acid, even in the presence of oxygen. Another adaptation is in the tumor's response to poor blood supply, which stimulates the expression of hypoxia-inducible factor 1, also known as HIF1. HIF1 is a transcription factor that upregulates transcription of multiple genes that control the synthesis of enzymes that are involved in glycolysis. Another adaptation comes from the combination of poor blood supply and preferential utilization of glycolytic metabolism in tumor cells. This combination leads to local acidosis, and this local acidosis in the tumor's microenvironment is believed to lead to increased invasion of tumor cells. These changes would be advantageous to the invasive tumor as they increase uptake and metabolism that promotes proliferation of the tumor cells. As a reminder, we will briefly explain how glucose uptake is regulated in other tissues. In adipose tissues and skeletal muscle, glucose uptake is increased in the presence of insulin. In the liver, glucose regulation is accomplished by glucokinase, which allows uptake of large amounts of glucose in the fed state. The anterior pituitary gland secretes various hormones that affect glucose uptake. One of these is growth hormone, whose secretions decrease glucose uptake in muscle. Glucocorticoids are secreted by the adrenal cortex and have an antagonistic response to insulin. Due to the poor blood supply of a tumor's microenvironment, glucose uptake is unable to be regulated by the above regulators. For Case 3, a 5-year-old boy is brought to the pediatrician's office and his mother states that he is sickly and often appears to be drowsy. She also states that he has persistent thirst and frequent urination. The pediatrician decides to do a glucose loading test. The patient's baseline serum insulin level was 0.8 and the level was 5.0 1 hour later after the glucose load. For reference, the control for the baseline blood sample is 6 plus or minus 2 and the control for 1 hour after the glucose load is 40 plus or minus 11. To begin, the pediatrician decided to do a glucose loading test due to her concern for diabetes. The pediatrician would be concerned for diabetes due to the complaint of persistent thirst and frequent urination, as these are often signs of diabetes. A person with diabetes experiences frequent urination in response to the excess sugar in the body. The kidneys cannot absorb all the sugar, and so it is excreted into the urine, causing fluid to be pulled from your tissues. This often leads to dehydration, which causes a persistent thirst. If one were to do a urinalysis on this patient, it would be expected that it would show a high concentration of glucose in the urine. With that being said, if the pediatrician ordered a fasting blood glucose level, it would be predicted that these levels would be above normal range. This is because the patient has type 1 diabetes, which means they have impaired glucose tolerance that is caused by a decrease in secretion of insulin. The decrease in secretion is due to destruction of pancreatic beta islet cells. Two regulatory enzymes that are likely to be active in this individual's current condition are pyruvate carboxylase and glucose-6-phosphatase. This is due to the fact that both of these regulatory enzymes are repressed by insulin. Due to the decreased secretion of insulin in this patient, these two enzymes are not being repressed and thus their activity is increased. Thank you.